Welcome everybody to the monthly webinar of EUROTOX. Today, the title of the webinar is Exposome Toxicology, combining different types of stressor. It will be given by Professor Robert Barouki. So Robert is a MD and PhD and is Professor of Biochemistry at University Paris-Cité and is head of the INSERM Institute of Health. I remember you that INSERM is the research institute concerned by health in France. His research is focused on the impact of environmental contaminants on human health, in particular the persistent organic pollutants and the endocrine disrupting chemicals, more generally on the links between the exposome and health. He is involved in several European projects, many European projects, the PARC project with linking exposure to health, the EELS and Neurosome project. This is about exposome. The ERA project, setting the research agenda in environmental, climate, and health. Oberon project, which is EDC endocrine disruptors testing. HIHEN, setting the agenda for human exposome research, and EREN, which is European exposome infrastructure. He also been involved in the networking of French and European research in the field of environment and health, as well as in communicating scientific data to citizens, and so he's one of the pioneers, I think, about the exposome. So thank you again, Robert, for your presentation. I remember to the participant that please shut down your microphone and your camera. It will be a 45-minute presentation, and then you will have 15 minutes to ask questions. If you are kind enough, please put your question in the chat before, so it's easier for me then to ask the question. And Robert, the floor is yours.
Thank you. Thank you very much, Mark. And thank you for giving me the opportunity to discuss these issues here today with EUROTOX. And so there will be a lot of introductory material at the beginning so that everybody is at the same level on the exposome. And then I will give a couple of examples showing how interaction between different stressors is really something that is relevant for all of us. Well, let me start by stating that although I will be speaking about the dark side of the environment most of the time, I would like to stress that the environment is not only about accidents, air pollution, contaminants in food. It's also about the solutions, which are like green areas, blue areas, social interactions, nice social interactions. So it's there's a good side about the environment, and let's not forget that. I think it's really important to highlight this. But from now on, I would be mostly speaking about maybe the dark side. Let me also start with some different concepts that are people are dealing with these days, and basically that started with a different history. I mean, One Health, for example, started mostly with infections and zoonosis and things like that, in animal and human infection. But now One Health is much larger than that, and it's basically combining both human and ecosystem health. So it's becoming quite large. Global Health initially started also by focusing on all the different areas of the globe. But now I think also it is addressing a large number of different issues, and not only infections, but also much larger chronic diseases and the effect of the environment. Planetary health has really a new concept that has also focused on the human and the planet. And basically, it has stressed planetary boundaries that you have all heard about. But essentially, it's all about how the planetary environment can influence also human health. So basically, all these concepts are like converging actually to a sort of big, large concept about global One Health, planetary health, whatever you call it. And I think the exposome is also one of the very exciting concept that started. I will give you some background on the history a little bit later, but basically combining all types of stressors that can influence human, but also there's the eco-exposome on the ecosystem. So essentially the message here is that, yes, we have different concepts that started from a different place, but basically they are all converging and highlighting the fact that humans, well, live in their environment, and there is a common interaction between humans and their environment. I will get back to the exposome a little bit later. So if we speak about pollution, is pollution relevant? Yeah, this is a WHO statistics. It's a little bit old, but basically the new statistics are very similar, basically stating that the global estimated death worldwide are related to different determinants, but one of the determinants, which is total pollution, is probably the most important one, maybe or close to smoking, tobacco smoking, but more important than some infectious diseases. So we have to take care of this. Obviously, this is mostly related to air pollution, but that's because that's where we have most of the data. And basically in many other cases, we really lack data. So it's really important to stress that pollution is very critical for human health. Now, do we know enough? If we just focus on chemicals, do we know enough about pollution? If we estimate that we have like 100,000 chemicals around us, basically we know a lot about 500 of those chemicals. This is from the EEA, European Environmental Agency. 500 out of 100,000, a little bit about 10,000 and 20,000, and basically very, very little about 70,000. We will not do the same work on the 99,000 chemicals that we have done on the first 500. Certainly not. There's no time for that. There's no, and I think we will be using very often now AI and computers to just predict basically what these chemicals are doing, and we cannot wait for all the experiments to be done. Although we have to confirm some of the data that we are generating by bioinformatics. So yeah, we still have actually as toxicologists a lot to do. Let me go back to the exposome and remind everybody about the significance. It's actually a very wide concept. It was developed by Christopher White when he was the head of IARC in Lyon in 2005. And basically, it's he defined it as all life course environmental exposure from prenatal period onwards, and it includes different processes, internal, external, and lifestyle factors. Basically, all exposures are included. And why did White think that this is important to introduce such a large concept? And because he was involved in cancer epidemiology and basically thought that a lot was known about the genetics in cancer, whether it's constitutive or tumor genetics. But whenever you are trying to consider other determinants, basically it's sort of a mess. I mean, about diets, about pollution, infections, habits, etc., behaviors. So he thought that it would be good to put all those together in a single concept, so it makes it maybe a little bit easier to focus and try to unravel all these non-genetic effects. But there's another consideration. He says all life course. It starts really from the prenatal period, maybe earlier, all the way to that. So there is a time consideration that is extremely critical here. But after Chris Wilde developed the concept, actually for a few years, nothing really happened. And then other people have started to try to build on that concept. And Rappaport has really basically focused very much on the internal component of chemical component, actually, of the exposome, basically stating that if you analyze body fluids for small molecules, you can learn a lot about the exposures. Gary Miller, he's a toxicologist, and soon later defined the exposome as the cumulative measure of environmental influences. But importantly, he added associated with biological responses. He's a toxicologist, so it's not only about the exposure, it's also about the biological impact. So exposome is much larger than just a collection of exposure. It has also to take into consideration the impact. Get back to this a little bit later. And also the NRC has developed this eco-exposome concept, basically extending it from the human to all the environment. And basically they described it as the interaction between different organisms, human on one side and organism in the ecosystem on the other side. So much more focus on the ecosystem. And then, Price and Vermeulen et al. Developed, sort of chemical biological interaction as at the heart of the exposome. Network science very much involved here. And with Price, we find something called functional exposomics, because most people still thought about exposomics as simply the collection of exposures. And if we wanted to add the impact, the biological impact, or even the health impact, then maybe another word which would be functional exposomic would be more adequate. Exactly like we have genomics, where we study the genome, and functional genomics, the outcome after the genome. So the same way we have exposomics, a collection of exposures, and functional exposomic, the actually biological impact and health impact of the exposome. And with Paulo Vines, we have focused on the socio-economic aspects and basically on the trajectory of exposure over life, and with a focus on how much the social aspects and socio-economic aspects are so important. So this is actually the history of the exposome, starting with wild, as I said, nothing much in the first year, and then a bunch of things happening much later. I will not go along all what is happening, but basically, as Mark mentioned, some of the activities now going on, the IHAN, which is the International Human Exposome Network, aiming basically at providing the European Commission with what we think would be important as a research agenda for the next years in terms of exposome. And interestingly, the NIHS in the United States had launched also a call for a sort of centralized coordination of exposomics, and I think the Colombia team won that one. And basically also they will try to advise on how to develop the exposome. So it's happening in different areas of the world, and we are getting close to what I think should be a sort of global human exposome program worldwide, exactly like we had the human genome program, although much more complex for the exposome, but I think we are heading this way, and it's really important that we have this very much international cooperation. I can discuss this a little bit later if you want. I have been very much involved in toxicology, as you know, and in exposome, and I'm a strong believer that toxicologists should get very much involved in exposome work. I will give some example a little bit later, but basically how we could be involved. First, exploring multiple stressors. I mean, we are very often dealing with a single chemical in toxicology. Now we can deal with mixtures of chemicals. This is also part, in a way, of the exposome, but we can also deal with the interaction of the mixed exposure to chemicals, same time exposure to different unbalanced diets, for example, or to social stressors or physical stressors. I think what we can do is start to really study the interaction between different stressors. And this we'll get to back to this in the second part of my talk. The other important aspect is long-term effects. Exposome is about all life exposure. So what happened in the uterus and during the fetal period will influence what our life will be and our health will be several dozens of years later. And this is something that we should address in toxicology, and we can address in toxicology. Also, bridging the environmental threats and human threats is something that is extremely relevant for us nowadays. And we as toxicologists always have to think about the regulatory implication of all this. So there are a number of ways through which toxicology can get involved in exposome research and be actually central. And this is basically what I'm showing here, how epidemiology, exposure science, statistics, data science can help us identify the stressors and their outcome, but how toxicology will be influential in basically addressing causality and supporting the work that is done in the other fields like epidemiology, all this leading to regulation in public health. So this is a very traditional scheme, but that's, I think, extremely relevant in exposome research. Another point is that we are now also, as I said, dealing with different concepts or frameworks, and many of you have heard about the adverse outcome pathway. I don't need to state it, starting with a molecular initiating event all the way to the adverse outcome. But we also, as toxicologists, know what ADME means, basically, the kinetics of what's happening in our body. And exposure scientists are now using this framework of aggregate exposure pathway, the different steps in the exposure leading to the internal dose that we are dealing with. So all these are linked together, tend to call them very often the AAA concept, bringing all these together. I think it's really also important and very relevant to exposure and exposome. Another point, as I mentioned, the exposome is all life exposures, and but before even the concept of the exposome was there, we had another concept called developmental origin of health and disease, DOHAD, and there are international societies in DOHAD. I contributed some time to those initiatives, basically stating that there is a vulnerability period during development in which exposure at that time could influence health much later in life. And this is completely in line with the exposome concept and should be basically incorporated. And we shouldn't distinguish anymore between these things. The important thing for us is what is the mechanism, and I will get back to this later. So now there is a sort of tradition in different areas of medicine, health effects, to have what we call the hallmarks. It started with the hallmarks of cancer, and actually in 2002 by Hanahan and Weinberg, and it continued with a number of other fields of medicine. And recently, Annette Peters and her colleagues published in Cell something called the hallmarks of environmental insults, just summarizing what is known until that time about basically what characterizes the effect, the biological effects of exposures. And basically you can see genomic alteration, epigenetic, mitochondrial dysfunction, endocrine disruption, nervous system disruption, oxidative stress, microbiome, a number of different effects. I took the challenge and added some other effects that I think are extremely important. For example, immunomodulation. I think this is extremely relevant these days, and you cannot ignore it. It is certainly part of the hallmarks of environmental effect. Cardiovascular, not enough studies on that, but should be more studies, and stem cells effect. But this is, I think this maybe should appear sometimes later as a second version. Okay. So as I said, long-term effects is a major point, and this is mostly explained these days, although there are different mechanisms possible, by epigenetic marks, since exposure can lead to epigenetic regulation, for example, methylation. And this could lead to something that is silent to begin with, doesn't appear in terms of health effect, but that can appear many, many years later as a health effect. One example would be the epigenetic regulation of certain genes in the adipose tissue that doesn't lead necessarily immediately to obesity, but over the years could lead to obesity. Now, epigenetic marks could be simply markers. They may not be an intermediate between the exposure and the health effect, and that's fine too, because they can be used as marker, actually, as relatively stable markers of exposure effect. But the mechanistic aspect is really critical, and also this is something we have to keep working on. I think epigenotoxicity should be a major topic for us in toxicology. And nothing happens actually in a field if you don't have the methods. And I have to say that mass spectrometry has been one of the major methodologies that have had the real development of exposome research, but also detectors. These satellites also and personal detectors are extremely important, wearables for this. And obviously, DOHAD was mentioned. Artificial intelligence is really critical. We have given some examples such as in text mining and interpretation that can really be used. PBTK, I don't have to insist on this. New approach methodologies like new systems. QSARs that are now developing, I think they will develop at a much faster rate now that we have basically the AlphaFold process where we can predict protein structure relatively easily. Adverse outcome pathway and DOHAD were already mentioned, though these are some of them. I mean, they are not all of the technology, but a concept or a field can develop only if the new methodologies appear and develop. So let me give you some examples about first why I started getting involved also in exposome and also the importance of the interactions between different type of stressors. I have worked a lot of time on the aryl hydrocarbon receptor or dioxin receptor. And basically, when I started many, many years ago working on that receptor, it was part basically of the detoxification system, basically detecting xenobiotics or contaminants, whatever, and inducing the xenobiotic metabolizing enzyme and transporter system, essentially leading to the elimination of compound. Very classical, very traditional. It was one, participating in detoxification. And then we found mechanisms through which this receptor also is involved in actually in toxicity, which seems paradoxical, but not that much. But I don't have the time to develop this today. But now, if you look like in 2024, this is not only the receptor for some, contaminants like dioxin or PAR or PCBs, but it is also a receptor for a number of endogenous compounds, basically derived from tryptophan, for example, or other compounds, dietary compounds such as polyphenol, and several microbial compounds. All these are involved in the aryl hydrocarbon receptor, leading not only to detoxification or toxicity, but also to immune effect, vascular effect, liver effect, myelin, immune system, with a number of effects on the phenome and on the pathology. So this was initially a receptor for a class of xenobiotics, and suddenly now it's actually a receptor for a very large number of different types of compounds with different functions. And I think this is typically a good example of what we try to call the exposome receptor, combining different effects and therefore the interaction with different types of stressors, for example, microbial stressors and xenobiotics. That was one example. The other example I would like to illustrate is possibly the interaction between unbalanced diets, for example, fat-rich diets, and compounds like dioxin. So we, in this study, it was a mouse study where we have treated mice either with nothing or regular diet, or with dioxin, or with high-fat diet, and the combination of both, and try to look at the liver, what's happening. And I'm not sure you can see very well, but you can see there's much more red things here, meaning that there is a lot of fibrosis, serious red, and there's also much more fat in this liver here, treated by both high-fat diet and dioxin as compared to the control. And essentially, maybe there's additivity or a little bit of synergy. I cannot say it basically based on the number, but maybe it is synergistic. So the combination of different types of stressor, having an unbalanced diet and having dioxin, can lead to health effects, actually, and biological effects that are much more important than each one of the stressors alone. And I think this is very much in line with the concept of the exposome, trying to study the larger picture of the exposure and not only focusing on a single one. Another example that I would like to set is also related to dioxins, but and a number of POPs that are related to dioxin. Let me give you some background on these experiments. It actually started several years ago, when we have noticed in cell lines, doing transcriptomics after treating the cell lines with dioxin, we have found that there were a number of genes involved in cell-cell interaction and cell migration that were induced by dioxin. And this was a surprise. This was not really known. So, and basically we then showed that indeed dioxin tended to separate cells from each other and to increase the mobility, the speed of the migration of the cell in vitro. And based on that, we thought that if this is true, those were actually tumor cells, MCF7 and the HepG2 cells where we did that work. So it was in cultured tumor cells in vitro. And we thought if this was true, then maybe these compounds may have an effect on the progression of cancer, not only on the initiation or promotion of cancer, but also on the ability of cancers in humans to lead to metastasis. And this is basically what we tried to show here. it was where we have the experiment. It's not an experiment. It's actually a clinical assay where we had women with breast cancer, and during the surgery, we of course took the breast cancer, did some transcriptomics on the tumor, and also had some of the fat that is around the tumor, surrounding the tumor, sort of micro environment of the tumor. And in that fat, we assayed for a number of POPs, including dioxin, and basically we then correlated the presence of these contaminants, pollutants, with the presence or not of metastasis. So basically, we found that when we combine, it's here we have the effect of mixtures of some of the compound of these contaminants, which had already, we know that they had a positive weight in metastasis, but we had this mixture, and you can see that it is at least significantly here correlating with the capacity to lead to metastasis. Other of those compounds are not correlated. And this is here on a metastatic risk. Here it's on the tumor size. We have a similar effect. So basically what this small, I mean, I want to stress that this is not a foolproof, because this was approximately a little bit more than 100 women. So it's not like this big clinical trial. We are now starting one, but showing that there is a correlation between the presence close to the tumor of these mixtures of POPs and the metastatic potential of the tumor. So I think this is relevant because, as you know, people die from metastasis mostly. I mean, and that's what kills people when you have cancer. But then we went a little bit further just to try to show whether it was important to have this microenvironment with fat cells. And basically what we are showing here, and let me just look at this column here. These are cells that are not, actually MCF7 cells that are not treated, breast cancer cells, not treated by anything. Then we treat them either with dioxin, or we add to these cells at pre-adipocyte, because this is what we are mimicking the environmental, the tumor microenvironment, adding these adipocytes. And then we did both, and we measured a number of different things. The cell numbers decreased, but then you can see that giant cells appeared that you can check here. Sometimes they had several nuclei, and this is correlated with the sort of aggressivity of these tumors in general. So we found a number of phenotypic correlates in culture, showing that a mix of dioxin with pre-adipocyte leads to this kind of phenotype in cultured cells. We went a little bit further and did experiments in zebrafish, in which we took tumor cells, treated them with either dioxin alone or pre-adipocyte alone, or the mix of two, injected them in the zebra, and looked actually for metastasis. The metastasis here are, you can see those dots here, and this is in, those sort of yellow dots in the, where I'm showing it with the pointer, and they indicate metastasis. And importantly, what we have seen is that only when we have the combination of the cells, tumor cells treated by dioxin and by the pre-adipocyte, only on those conditions we can see brain metastasis of these in these experiments. This is from zebrafish. It was really extremely clear and very interesting to see that we can reproduce metastatic potential by pre-treating the cells with both dioxin and their sort of adipocytic microenvironment. So all this goes together, and we built, based on that, an adverse outcome pathway linking the activation of the AHR by dioxin all the way to breast cancer metastasis, and based on some of the observation that we have made during our different types of experiment. This may not be a complete AOP, but at least it's a start, and we published this a couple of years ago. So this was the story about, interaction between the human microenvironment, the adipocytes, and dioxin. Another example of important interaction is xenobiotics with microbiome interactions and different type of stressors. And it can lead to many, many different things, including bioaccumulation of xenobiotics, growth inhibition of the microbes, growth promotion, changing in the type of bacterial metabolites. There is also reported an impact on the virome and on xenobiotic metabolism. So the interactions are multiple between the microbiota, which is also responding to many, many other clues, actually, and the exposure to xenobiotics. And it can go actually both ways. In one case, with the metformin drug, it actually is beneficial to have this interaction with the microbes. And for case of glyphosate, it may be actually toxic to have these interactions, as this may change the constitution of the microbiome. This would be one of the mechanisms of action of glyphosate. At least this was published a couple of years ago. And there are very good examples. I don't know if you know the story of melamine that has been used in milk in China. It was toxic to the kidney, but melamine itself is not. It has to be degraded and metabolized in the microbiome, intestinal microbiome, leading to cyanuric acid, which itself is actually toxic to the kidney. So these interactions are multiple and may lead to a really significant effect. Another point that I would like to stress, and getting out of the chemical or biological field, is an example of what can be done on basically reproducing in animals social stressors. And here, the experiment, actually, you can see it here, I hope, on your computer. You can see two species of rat, one that is actually a very nice rat, it's not aggressive, and the other one is an aggressive species of rat. We are describing here what we call the social defeat experiment, where you expose that rat to the aggressive rat for 15 minutes every day for example, a few days. And then at the end of the day, this nice rat gets completely depressed, and basically they don't move anymore. They are really in not so good shape. Even if they are separated, I mean, they don't move anymore. We call it social defeat. And accompanying that, and very interestingly, assay BDNF in the serum, brain-derived neurotrophic factor. It leads to a strong decrease in BDNF. And some of those rats recover normal BDNF and basically are okay. Others do not recover, and they are really not in good shape. And if you add another stress, whatever it is, not necessarily social. So here you have a sequence of stresses, not stresses at the same time. But another stress, oxidative stress induced by some chemical or whatever, then you have really a very bad situation for those rats that have still a low BDNF, and it goes to the pathologies and death. I think the nice thing here is that there's a correlation between the decrease in BDNF and the social defeat state of the rats, and it seems that BDNF is very critical for how good the rats are recovering. And I think the important point also is that BDNF is a very well-known target of chemical stressors. I mean, we've done in the HBM4EU program, we studied the effect of bisphenol A on BDNF and showed also that it was could be an effect biomarker of bisphenol A, and we showed that it was at the epigenetic level. So clearly here we can combine, and this is combined here in the sequence, different type of stressors, social stress on one hand with like chemical stress on the other hand, and to have an effect in the long run on morbidities and death. So those were a number of examples of interaction between different types of stressors, very relevant to the exposome in toxicology. And just to give you a flavor of that, how important this is in the clinics, we took the example of liver and liver pathologies, starting with steatosis, fibrosis, leading ultimately to cirrhosis and ultimately cancer. And here you have all the stressors that are involved in liver effects. And there are a number of them. For example, obviously alcohol, I mean, diets, but also viruses are extremely important. Some drugs are hepatotoxic. And there are good examples in the literature that we have actually summarized in this article, showing that the interaction between, for example, some mycotoxin and some viruses is really more than synergistic to, at least synergistic, leading to this liver pathology. And there are a number of other examples of interaction. I mentioned the microbiome with drugs, with xenobiotics, etc. And even air pollution actually is now known to lead to this effect through a mechanism that has still to be completely determined. So this is an example of a pathological state where we have these interactions between a number of chemicals and leading to pathology. Let me just spend a couple of minutes on other issues, and we have to be involved in global change effects and climate change or biodiversity one way or another, because these are becoming extremely important issues, and there's no way we can avoid being involved in this. And actually, many years ago, we published a small paper in the Lancet. It's a commentary stating that focusing on health can save maybe the negotiation on climate change. Here, there are now going on some negotiation at the COP in Azerbaijan, and it's really important to put health in the top priorities. It wasn't the case in 2015 when we published that paper. It's become the case more recently, actually. And this is a small attempt. This is a very traditional picture that you can see in many climate change reviews, all the stressors that are involved, and there are many of them, and they are acting at the same time, mixtures, exposome. I mean, climate change is simply one case of modifying the exposome. And below, you can see all the health effects that can be related to climate change, infectious disease, including mental health. This is an attempt to try to find the biological events, the intermediate. And I think we as biologists and biochemists and toxicologists, we can do that. I mean, we can help putting the intermediates between these exposures here and the associated health effects there. And this is an attempt. It's certainly not perfect, but I think we have to keep working on this to be involved in this important field. Another field is the loss of biodiversity. Not going to detail all the observations that have been done showing that there is a major loss in biodiversity worldwide, and there's an almost extinction of certain species. And here also, let me skip this. Yeah, here also we can also play a role in trying to identify the mechanisms through which the increasing or decreasing biodiversity could lead to positive or negative effects on health. This is a nice example of what we can call a clean environment to the left, a clean but toxic environment to the skids. And here you have maybe a dirtier environment, maybe with some significant advantages, mostly on the immune system and some immune diseases, allergic diseases, or immunomodulation, or autoimmune disease. And there's an attempt here to show that epigenetic may be involved in these mechanisms, linking the loss of biodiversity all the way to health effects. So this was an attempt, but much more needs to be done here. I think we still need some more work. So let me try to summarize and open the discussion maybe. So why would the exposome be useful? Why should we as toxicologists be involved? Well, it's a better integration with many disciplines, like in epidemiology, exposure science, bioinformatics, maybe AI in the future. We have our role in determining the causality. We can link our effects that we observe to actual exposure and therefore contribute to risk assessment. We can better understand the mechanism of various types of stressors, not only chemicals, which we do most of the time, but I think we can contribute to the mechanism of other stressors, being involved in mixtures, obviously, and interaction with different stressors, addressing vulnerability, in particular during the early phases of life, and addressing long-term effects, for example, through epigenotoxicity. There are a number of regulatory implications for, the good example is mixtures. How can we deal with mixtures, or how can we deal with the combination of toxic compounds with, bad diets, and how much this can be important. Vulnerable population, obviously, we have to, I think the Exposome puts them into under the lights, and public health messages in which we do not only focus on a single type of stressor, for example, a chemical, but we try to have a sort of much larger vision so that the citizens do not hear contradictory things. For example, somebody telling them that they need to eat vegetables, which is, fruits and vegetables are really good for their health, and some other toxicologists telling them that, well, most of them are full of pesticides, traces of pesticides, and that may not be so good for the health. So we need to have a consensus here and provide a single Exposome-inspired communication to the public. More precision prevention, if we can test and assay a large number of things, and here chemicals or maybe even social stressors can also be organized in different type of determinants, and we can do that. And maybe ultimately some score when addressing the toxicity of what is coming with the food. And food is not only about nutrients, it's a lot of contaminants too, and maybe having a score identifying this in the future, this could be extremely relevant in toxicity. With that, I would like to thank you, and I hope we can still I think we still have time for discussion, Mark. So I will stop the sharing if you want, so that I can see you better.
Hello. Thank you, Robert. I'm sorry there is a phone behind me, but I hope it's going to stop. Yes, thank you very much for this very, very comprehensive presentation about the Exposome and everything about Exposome. So I already have some questions from the chat. So I will start with a question from Baron. Would the stressors always function in synergy, or in some cases, presence of one stressor can inhibit the role of other stressor in either a positive or negative trend?
Oh yeah, I mean, this is obviously, we should not have the synergy. Many people actually do not believe so much in synergistic effects. They believe that at best we may have additive effects, but so it's a case by case. There's no rule, absolutely no rule, and there shouldn't be any rule, but it's a purely case by case study. You have to show, if you think there's synergy, you have to show it. If you think it's additive, I think the base, if you have different stressors having a similar biological effect, the best maybe guess would be additive effect. But in some cases, it can be opposite effects, and then you can maybe relieve some of the effects of one stressor with the other. So I don't think there's a general rule for that.
Thank you, Robert. So I have a second question from François Busquet. That's a complex question. How to translate Exposome concepts into health legislative European action, EU action?
Okay, it's again a case by case problem. The Exposome is such a large concept that it includes many different areas. I think the first thing is mixtures is a good example that we should really now focus on mixtures and try to find a way of taking mixture into consideration for regulation. That's one example. The other example would be, for example, focusing on regulation on food and taking into consideration the fact that food are not only nutrients but also contain contaminants, and that therefore it's extremely difficult to give an advice only based on the nutritional aspect and not taking into consideration the toxicological aspect. I don't know how this could go into legislation. I'm not really, but I think, having in mind that we are not dealing with a single effect alone, but with a single stressor alone, but maybe with a mixture at the same time, should lead the legislator to be very cautious in certain fields. Yeah.
Question from Gad-Olivier Arfi, sorry. Thank you for a great presentation. Could you please comment on microbiome dysbiosis and immune system disorders linking to exposome, especially linking to allergies like food allergies.
Yeah. Well, I mean, this is a big chapter now, becoming a real big chapter in pathophysiology and toxicology. And I'm not gonna, it would be too long, but just take a small example. I mean, I talked a lot about the AHR because I'm too very much focused maybe on that. And the AHR is in the intestine. It's in the immune system, actually, of the intestine. It binds a number of microbial metabolites coming both from the regular, normal, bacteria present in the intestine, but also of some other bacteria that are, increased during certain pathological condition. And what AHR is doing is, on one hand, allowing the tolerating these, some of these bacteria, and on the other hand, giving a relatively toxic message to the immune system. How this leads to allergy, I cannot comment. This is far from my field now, but I'm just commenting on the fact that the same receptor may be involved both in xenobiotin on one hand and also in microbial metabolites, and this could lead to real very strange interactions.
I just can complete the answer from Robert that there is at least one, maybe two papers showing that dysbiosis in rodents is involved with an augmentation of skin allergy in a model of skin allergy.
But the truth is that dysbiosis is involved in so many different pathologies. I mean, nowadays that we have to, at one point, we have to clarify this one way or another.
Yeah. There is a question from Emanuela. So thanks for the presentation, very informative. If we think about substance regulation, this is done substance by substance. How from the exposome can we identify the substance of concern?
Well, I mean, you can, people have tried to do that. For example, in epidemiological studies, they have been assaying not only one substance, but a large number of substances. And then based on statistics, they have identified the mixture of substances that are most related, for example, to effect on neurobehavior or neurological effect on one hand, or on metabolic effect on the other hand. So they know what is the mixture of substances that is mostly correlated with that. Based on that, they can also, based on some statistical studies, they can also tell from these mixtures which are the most critical chemicals. I mean, in this, chemicals are not equal in a mixture. I mean, so some are more equal than others, if I may say. So, and they're identifying this may also support regulation, both on mixture, but even on certain chemicals by themselves. So I think this is promising one way or another.
There is now a question from Saed. Thank you very much. Very good information was provided by the master. Okay. So we have a fan here. So how certain is the impact of external exposomes, such as climate change, that's a good question, on internal exposome, such as oxidative stress, and can it be trusted?
Well, I mean, our body is the reflection of our environment in many ways, with the influence of genome, of course, but it is a reflection. And sometimes, well, first, the external chemicals, you can get them inside. And this is sort of what we call the internal component of the exposome, but we are also dealing with the impact of these chemicals in the internal. And oxidative stress is clearly a good example, but it's a very poorly specific effect. I mean, so many different stressors lead to oxidative stress that it's not so specific. So can we really correlate oxidative stress specifically with this or that stressors, knowing that people are exposed to many different things? In some cases, we have been able to do that. People have been able, but it lacks specificity, but it's clearly very much responsive. And, for air pollution or eating contaminants or even imbalanced diet leading to oxidative stress. So yes, we can find correlation. I hope that it's nice to have oxidative stress or inflammation, but these are very poorly specific. They're just a sort of danger. I think it's a signal for danger in a way, like we have it in the immune system. But now we have to go a little bit further, and I'm really hopeful that epigenetic will provide more specific signals about the internal exposome being influenced by this or that stressor.
Yeah, just so I wanted to extend a little bit on that because I had also a question. Because, for example, for climate change, if you can identify some biological process that are modified by climate stress or some other stress, it means that you can study the interaction and you can maybe start to do some risk assessment on that, no?
For climate, you mean?
Yeah, if you identify that climate can be associated with some modification of biological processes, and you know that some chemicals are also affecting these biological processes. So
yeah,
I
think yeah, you're right. I mean, that's the aim of it. I mean, that's why I think, I was asked to do this, to write this paper by one of my colleagues who wanted me to say, what can toxicology say about climate change? And he's an epidemiologist, and it wasn't clear. And it wasn't clear to me either when I started. So I took this really picture where you have all the stressors and all the health effects. And this is, you can see it in many different, in the Lancet Commission, in many, many different cases. And I tried to put in the middle, the biological effect from what I knew. And I may have missed very important things, but I think it's an important exercise for us to do, because that's where we can be, we can influence this knowledge, and we can maybe start to find biological markers that are extremely relevant, maybe predictive of something, maybe interaction with some other things. So I think it's an important exercise. I'm not sure I've done it in a really comprehensive way. It's just a start, but it would be nice if a group of people would sit down and try to build this picture in a much more detail and more references, actually.
And so I have a last question on my side. I have a tarte à la crème question. It is the tarte à la crème question. This means it's a very general, and I think it is okay. You have different type of input. Okay, you have input of the effect of the chemicals, maybe the input of the climate, maybe the input of some of the obesity. And so, as we know, artificial intelligence is going to solve basically all the problems in the world. And we know that artificial intelligence is here to integrate different endpoints like transcriptomics, proteomics, metabolomics, etc. And so we know that it has started to be used for that, and it's working, but it's beginning to work. So is artificial intelligence could be a start of an issue, or some people are thinking about using artificial intelligence to integrate some endpoints from climate, some endpoints from exposure to chemical and biological effects that are affected. So, for example, if you're living in Norway and you're exposed to that, and then the climate would modify, etc., etc.
Yeah. Well, I mean, I think there's a lot of promise in there, and we certainly, even although it's a cream pie, as you say, in tarte à la crème, I like tarte à la crème, and I think it's perfectly all right for me. Of course, we can say everything can be solved in theory. Now you have to do it. And we have used artificial intelligence, for example, for an intelligent text mining of the literature. I mean, in some cases, you have tens of thousands of papers on a single topic, and you want to extract some information. It's not just asking for one word, mining for one. It's really asking for two words, for example, bisphenol and cancer, and putting scores on the distance between the words and where they appear in the abstract, the beginning or at the end. I mean, putting some more intelligence in it, and this will lead to a real exploration of the literature that is, and of the data, much quicker than what we used to do. So I am really very much in favor of developing that to a large extent. I think we have to do it. It's not an option. I mean, we have to do it. And we should do it. And in several cases, in structural biology, for example, I think it's extremely promising.
Okay. So thank you very much for the answer. So we don't have any more questions in the chat. I don't have any more. And so we are arriving at the end of the hour.
Okay.
So we had 61 participants, which is a great attendance.
Okay.
And so thank you again, Robert, for the presentation. Excellent. Thank you for the question. Thank you for the audience, for the question. It was very interactive. And I wish you a very nice weekend.
Thank you. Thank you for inviting me, and see you soon.
Okay.
Bye-bye.
Bye-bye, Robert. Bye.